what is behind all these breakthroughs in ag genomics that we've been seeing over the past few years? In canola, in wheat, et cetera, et cetera.
I think a lot of the advances that we've seen in agriculture genomics and the sequencing of canola genomes, wheat genomes, pulse genomes is the result of a lot of innovation that started with the Human Genome Project in the 90s. And advances in sequencing technology and insights that our scientists and scientists around the world have developed are able to do these large genome assemblies. And plant genomes are huge compared to humans and animals because of the complexity of the genes, genomic structures in plants.
I think a lot of people are surprised by that idea, that a lot of people who aren't in the industry are surprised by the idea that, wheat, for instance, could have a much more complex genome than a human. And you talked a little bit about that, but can you sort of just give me a bit of an explanation as to why that's the case?
Absolutely. So, for example, bread wheat actually has 3 genomes, A, B, and D. Canola has 2 genomes, A and C. And again, this is a function of the evolution and development of plants where they become, polyploid. Duplication of genes, combinations of genomes. It's really part of the evolutionary survival, if you will, of plants versus animals.
Now, you mentioned that it's for evolutionary survival. What's the benefit of having that? Why do they need it and we don't, basically?
Well, I'm sure there's some evolutionary biology scientists out there that'll give you the right answer, but the way I've always looked at it, I can get up and run away. A plant can't. It's got to stand there and take it. And it needs the diversity and multiple copies of different genes to be able to have that resiliency. That's how I've always looked at it. It's probably completely inaccurate, but I think being able to run away from problems is a good thing sometimes.
Most likely, yeah, in an evolutionary sense, that's for sure.
One of the things that we're living through the pandemic, the emergence of the variant strains of COVID-19 is an example of how sequencing has been able to accelerate learning to understand what the risks are in increased infectivity of different variants of COVID-19 and develop understanding of whether the vaccines are going to be effective or not. So again, that ability to go from phenotype to genotype to understand what the risks are in human health applications is the same speed and insights that we need for improving plant varieties, animal varieties, discovering new tools and technologies in the ag space as well.
So if I'm understanding you correctly, we can draw a line from the sequencing of the human genome through to the agricultural breakthroughs we've seen in the last few years, and actually that same technology is in evidence with the rapid pace of being able to deal, relatively speaking, with the coronavirus.
Absolutely.